I'm really thrilled to be here today to share with you a bit of the things that excite me, that get me out of bed in the morning, that make my lab run. I've been in the Department of Chemistry for about 7 years now, and when I think about what my title is, I always have trouble with my affiliation. My job is a chemistry professor, but it's a very interdisciplinary lab. We have chemists, biophysicists, and applied physicists coming together to complement each other in this really interesting way. And so what I'd like to do today is tell you about the work that we've been doing. One of our prime motivations is thinking about bacteria cells, how they work, why they work. But really what I want to tell you about most of the time is the tools that we're building to enable these investigations at a very, very small scale. You all know that bacteria cells are tiny, and so we have to build new microscopes, and I'd like to introduce you all to these high-resolution microscopy techniques that we're using in my lab. So let me start by introducing bacteria and telling you a bit about why I got so excited about working with bacteria. And you should know that I come from more of a physics background, physics and chemistry, and I didn't know much about bacteria cells. And as my career has developed for the past 10 years working on these bacteriology problems, I've really been interested in seeing how bacteria are involved with every aspect of our lives, from ecology and the environment to problems of sustainable housing. And in my lab, we really think about two specific problems. One is human disease, and the other is human health. And so what I'd like to do today is tell you about the tools that we've been building in order to address a couple problems, and I'll end by telling you a few of the key discoveries and key directions that we're thinking about in terms of both human disease and human health. And when I say human disease and human health, what I mean is we all know that there's bacterial infections, that there's disease that are caused by bacteria, and one of the models that we use in my lab is Vibrio Cholera, the agent of the cholera disease, to think about how bacteria affect disease. But also in my lab, we're thinking about how bacteria help us stay healthy. And we also all know that our doctors are telling us to eat more fiber, and all over the media, it's telling us eat probiotic yogurt. This is because there are millions of bacteria that live in your gut and all over your body that help you maintain health. And so I'll tell you also about a problem that we're thinking about, which is that there's bacteria in our gut that digest complex fibers for us so that we don't have to do it ourselves and actually ensure that we'll stay healthy. So these are the two sorts of problems. The first is specifically, how does Vibrio cholera, this bacteria that causes the cholera disease, how does it make a toxin? And the second problem is, how does the gut microbiome, the bacterial communities in our gut, how does that help us, and how does that work? The problem, the really overarching theme in my lab, is this problem of size. And so what's important to think about is that these Vibrio cholera cells, if you look here at this scale bar, that's a ruler of 1 micrometer, 1 micron. These bacteria cells are about 4 microns in length. This is the Vibrio cholera cells. And this over here is, on the right, is the Bacteroides cells that are in our gut. They're all on the scale of 2, maybe 4 microns. They're very small. And just to situate you, a micron, or a micrometer, is about 100 times thinner than your hair. So it's really small. It's beyond the limit of what we could see with the naked eye. Just to think a bit more about the scale, I wanted to put up this Michigan-related graphic that was prepared by Liz Wasson over in the LSA office of the LSA magazine. Liz was talking to me about science communication and thinking about what does it mean to be so small. And so we thought about this thing, and I apologize, I know the size of this is small, but really, you know, the idea is that as we go smaller from things we know to things we can't see, it's hard for us to appreciate just how small everything gets. So what we're going to do is try to get down to the scale of nanometers, which is the size that we're after in my lab, and we're going to start from the scale over on the right of 100,000 nanometers, which is sort of what we can see with our eye. That's about 1/10 of a millimeter. And the way I like to think about this is thinking that if this millimeter or 1/10 of a millimeter is somebody walking, this is a good hike, right? Somebody walking from the LSA building over on Fletcher, walking all the way to the water tower in Ipsy. It's a good walk, right? It'll take you most of the day. That relationship coming down to the nanometer scale, so the distance we need to get to come from something that we could see to something we can't see, is equivalent to going from that walk from campus to Ipsy. It's equivalent to the ratio between going from that to just stepping over that little M stone at the center of the quad, in the diag, right? So we're really, really bringing it down a scale. And if we think about it getting bigger, it's a bit easier, right? Getting bigger, if we get 5 times bigger, we're already driving to Detroit. And if we get 10 times bigger, we have to get on a plane, right? We have to go to China. And if we get 10 times bigger than that, we have to actually go to the moon. So these increases in scale we could kind of picture as being outrageous. Getting small is actually equally outrageous. So thinking back to going smaller, the reason we're interested in this is because bacteria are so small. So remember that we go from the walk to Ipsy, which is the things we could see, and we're trying to get down to jumping over that 3-foot stone in the diag. Bacteria are really small. So here's a ruler that I'm showing you, and every time I go from one of these clusters to another, I'm going 10 times smaller. And I want to tell you that looking at bacteria is a special challenge, even compared to looking at human cells, which we all appreciate are pretty small, because human cells are more on the order of 10 microns. Bacteria cells are 10 times smaller, on the order of 1 micron. And then the structures inside the cells that I would argue really affect the function, those are even 10 times smaller. We go down to the cell membrane, the surface, the wall that makes up the cell, and then we could go 10 times smaller than that, down to the size of proteins, which are one of the things that really do the work inside the cell. It's hard to see things that are small though, right? So I told you already that the naked eye could see about 1/10 of a millimeter. The naked eye could see hair, and that's about the limit. So this is a graphic from one of my favorite websites from Florida State University that explains tons and tons of things about optics. And what they're showing you here is that the range of sizes that a light microscope can see range from experiments that I've done in my house with my kids who are up in the front row, right, putting a bug onto a microscope and seeing it. You could see a bug by eye, but the microscope magnifies things and we see it a lot bigger. We could go down to putting hair on there, and I've done things like looked and seen that my hair that I color blonde is actually thinner than my hair that doesn't get the treatment. If you go smaller than that, though, you're really getting to the limit of where the light microscope can be. And so the bacteria cell is right at that limit, and looking inside a bacteria cell is a new challenge. I'll tell you a secret. It's not going to be much of a secret anymore. This size scale is actually why I hated biology in college. So I was a chemist. I was a physical chemist. I took a lot of math and physics. And my problem with biology was this. This is a gorgeous textbook. So Campbell, it's beautiful. I still have it up on my desk. Beautiful, beautiful textbook, walks you through the canon of what we know about cell biology. And the problem is that the page about bacteria cells, and I just went and scanned this last week, it really shows a cartoon. It says that a bacteria cell is different from a human cell, and that's because it's smaller, and it doesn't have a nucleus, which is where the DNA is inside a human cell. It says that it doesn't have membranes around the different functional units. But beyond that, it really doesn't tell me much. It tells me that there's this sort of shape with some stuff inside, and that turned me off completely. And it wasn't until I had the opportunity to do biophysical research that I came back to biology and said, wait, no, everything is so interesting. Everything in there does have form, does have structure. The problem is exactly what we've been talking about, right? The problem is that my microscope can't see anything that small. So this is the cartoon that I came out of college with, and this is sort of the baggage I came to my biophysical research career with. I had this bias that, oh, bacteria cells, they're just a Ziploc bag full of stuff. And I have been convinced over the last 10 years that this is 100% not true. All this stuff is working together in an intricate way such that the placement inside a cell matters, not just if it's there or not, but is it at one end or the other. I've been convinced that the timing as the cell grows is super important in terms of controlling cell viability, in terms of controlling survival of the species. I've really been convinced that this cartoon is just a starting point. So let's start to look into that, and the way that we start to look into that is with a microscope. And so this is an example of the first microscope. This is from Robert Hooke, who was a British scientist back in the 17th century. And this microscope's actually on display still at the Royal Museum in London. I love a few things about it. One thing I love about this microscope is that it's leather coated. It's perfectly embroidered. It's just so aesthetically pleasing. But the other thing that's amazing about it is that this is years and years ago. This is before quantum mechanics, right? This is before cell biology was really around because we couldn't see anything very small. And this microscope was enough, engineered by hand. All the lenses that go inside were engineered by hand. This microscope was enough to start seeing inside of traditionally inaccessible materials. And so this is the real first micrograph that was published, and it's a picture of cork. So, you take your bottle of wine, you pull out the cork, and you look at it in the microscope, and this is what you'll see. And Hooke actually coined that term cell, not in a biological system, but actually in this cork. He looked at the cork and he said, look at these little subunits, right? Each one has this really nice structure. This is why the wine can breathe just right, and this is why the rubber corks on your wine actually have to be very carefully engineered. Robert Hooke looked at these, and he actually said, these little cells, they look like the monks' chambers in the monasteries, and so he named these cells after the monks' cells where they were living. And so I just love this idea that there's a bit of a Renaissance man type of feel to the story. So our microscope today is not leather, it's not embroidered. It looks like this though. But the nice thing is that it is super accessible. We look into it in our eyepieces, or actually in my lab, we almost never use the eyepiece. We just hook this up to a camera and we look at it on a computer screen and we record everything we're seeing in real time. We look at our sample that's down in the middle and we magnify it exactly the same way that Robert Hooke did hundreds of years ago. And so the microscope is a really, really powerful instrument. We love looking at it. We love the fact that we could take bacteria cells while they're still alive and put them onto our stage here and watch the cells grow and divide, and then look inside the cells and watch how the components are moving while this is all happening in real time. It's very accessible. We can add chemical reagents and see that all in real time. However, there's a limit. So let's start thinking about what that limit might be, and the limit is light. So a light microscope is really exciting. We all know that even in elementary school, we can have light microscopes. In my dining room, I have a light microscope. We could put any sample on there. We could put live samples. We could put complicated samples. But we're still using light. And the fundamental limit of light is that it's a wave. And so many of us have heard about how light is a wave. And when I mean that light is a wave, I mean that if it's traveling from left to right across this screen, there's crests and troughs, and there's some specific size. So the distance between two of those peaks on that red wave, that's the wavelength of light. And for red light, that's about 600 nanometers. So that's pretty small. That's half of a micron, right? So I told you my hair is 100 microns. The bacteria cell is 1 micron. The wavelength of light is about half a micron. So we're down at the scale of these cool bacteria cells. So we have our red light. We can also have green light. We can have all the colors of the visible spectrum. The green light has a shorter wavelength, as you see up here. And so the peak-to-peak distance, the wavelength of the green light, is a bit smaller. It's about 550 nanometers. So we're going down from 650 nanometers to 550 nanometers. Again, right, we're still skirting this area of half a micron. We're skirting this area of the size of a bacteria cell. We could think about the light this way. Here's those sizes again. This is the all of the radiation that you might see in the universe, going from very, very small, the gamma rays at the left, up to very, very big radio waves at the right. The radio waves are at the end, and that's why if you drive through a tunnel, you don't hear your radio anymore, but you might still get, you might still see some light. So the radio waves and the gamma rays span end to end, this big, big area, but we're restricted in optical microscopy to that middle region, about half of a micron, about half of the size of the bacteria cell. So light is a wave, and it's going to put a fundamental limit on our microscope because we can't resolve things that are much smaller than the wavelength of light. So I'd like to do a demo and think a bit about how the microscope limits our diffraction. So if we could turn down the lights. What I'm going to do here is what's called the single slit diffraction experiment. And what I have over here is a laser in the center, and I'm going to send it through a very narrow slit. And the most interesting thing happens. The light has a wave nature. This experiment was done before people knew that light has a wave nature. This was part of how they figured out that light had a wave nature. And those waves are going to interfere with one another. And what I'm going to do is send that interference pattern to this mirror on my right and then over to the wall on the left. So y'all can look at that wall. So let me go ahead and turn on the laser down. So what you see over here on the right, I had a laser. It was just a perfectly symmetric object. And what you see over here on the wall is that there's this peak on the wall with a peak width. And then there's some small features out at the end. And the distance between those features is going to be the smallest distance we can resolve in a microscope. And that pattern at the center is the one I want you to remember as I move forward. So if we can put the lights back on. Screen. All right, so this pattern up on the wall, I've reproduced it on the right side of my slide. So I'm going to go ahead and turn off the laser. And what you see over here on the slide on the right side is that diffraction pattern. I've turned it sideways now. So what you see is that I took a laser that had a wavelength over on the left side and sent it through a slit that you see in the middle of my screen. And what we make is that pattern on the left side on the wall. What we make is this center with a lot of intensity and near it a trough. And so even if I have a really infinitesimal, tiny little object that I'm trying to image, this slit has given it a finite size. So this experiment showed us that light has a wave nature, but this experiment is also very important in microscopy. And the reason for that is that the microscope actually acts like a slit. So here's that pattern that you saw before again. You see the focused light at the center. It has this finite size. And then you see some fringes at the bottom and at the top, those pale gray stripes. And light, the microscope objective and the microscope aperture actually serves as the slit in that single slit experiment. And so what happens is that we actually see this pattern in 2 dimensions. So this is a 1-dimensional pattern, just a straight line. The microscope actually looks like this. So it gives you a circular pattern, but it's the exact same thing. The image of even a really, really tiny molecule, even a really tiny dye, is going to look like this. There's a finite size at the center, and then there's some fringes out to the side. This is the fundamental limit of microscopy. So the rule of thumb is that because the image of anything, no matter how small it is, a thin bacteria, a protein, a dye, anything really, really tiny, it's going to look like this. It's going to have a finite size, and that size is related to the wavelength of light. That size is about half a micron, like that wavelength of light. And what happens is, this is an image of many, many dye molecules scattered around a surface. If those molecules are far apart, we could see them. As they come closer together, they start blurring out. And so up at the top, for example, where it says unresolved, you see that there's 2 dye molecules that have come so far together that those patterns, like the stripe that you saw on the wall, they're overlapping and we can't resolve them anymore. So when you say my microscope resolution is not very good or my microscope resolution is a micron, this is that size scale that we're thinking about. We're thinking about how far apart can items be in order for us to resolve them. And this was discovered, another sort of older story. This was discovered by Ernst Abbe, and there's actually a memorial to him in Jena in Germany. This says Ernst Abbe, and I could blow it up. It actually has on this circle his equation in his handwriting reproduced from his notebook of this process. So I've told you so far that we want to look at small things, that our microscope can look at things down to a certain size, and that the light microscope is very, very flexible. And I'd like to tell you one other reason that we love the light microscope, and that's because of fluorescence. So one problem that we have is something like a human hair that we're all pretty comfortable. We can put on the microscope. It's kind of tough to see. So I always will do demos and things like that for middle schoolers, and my students will grab some things around lab, and you always pull out a piece of hair and throw it on the microscope and look at it. And when we look at this, it's kind of hard. We have to squint a bit. We have to get the focus just right, and the contrast is very low because one hair does not absorb a lot of light. It's a very small object. So one hair does not absorb a lot of light, and we say that there's not a lot of contrast. So the way that we can attain contrast, even if we want to see something very, very small, is with fluorescence. And what I mean by that is I want everything to be dark except for the item I'm looking at. I want the signal to noise to be very high. I want the contrast to be very high. And the way that I do this is with fluorescence. And fluorescence is a really powerful tool, and I want to tell you a bit about how fluorescence was discovered, or how the process of fluorescence was discovered. Another one of my favorite chemistry stories. This is a scientist named Stokes, and those of you who are more familiar with fluorescence might know that there's something called a Stokes shift involved in the fluorescence. So there was a scientist named Stokes, and this is what he was doing. This is another one of these great, you need a beard and you need to stroke your beard to do this experiment. He was in the church, right? As everyone is, right, sitting there thinking, and he's holding up a glass of wine. Why not, right, if you're going to be doing philosophy? So he's holding up a glass of yellow wine, or white wine, right, it's yellow, and there's a solution of quinine, which is a nice chemical that happens to be fluorescence, and what he's doing is standing in the church, looking up to a stained glass window that's letting in only blue light. It's a blue stained glass window. And he's stroking his beard. He had to have been, right? So he's stroking his beard, and he's holding up his glass of white wine. And he looks at the quinine, and he sees that the solution of quinine is glowing. And what he actually did in this way is the first fluorescence experiment. What he found out is that if we excite quinine with blue light, we can't see the blue light through our yellow wine glass, but we can see the fluorescence from the quinine through that. So we're seeing these changes in colors. So fluorescence in general is this idea that we have some dye or some molecules or these things called quantum dots, or in my lab we use something called fluorescent proteins that are fluorescent. And we can excite them, we can shine them, illuminate them with one color of light, and we'll see emission at another color. And so what you're looking at here are some typical dyes, and they range from having a blue color to a red color. If we excite them with ultraviolet or violet light, we take the electrons in those molecules and we excite them, we give them more energy, we put them into a different state. And when those electrons relax, there's conservation of energy and a photon has to be emitted. So the photon's emitted, the light is emitted, and that's our fluorescence. So if we could turn off the lights, I'd like to do a demo. Thank you. What I have here are not dyes, but they're quantum dots. They're another type of label we tend to use. And what I'm going to do, I'm okay with it dim. Or is that's as low as it could go? Great. What I'm going to do is shine some light on them. So you see that the one on the left is yellow. The one in the middle is a sort of brighter yellow. This guy is green, and this guy's a different green. And I just want to focus on this again. I turned off my light. You don't see anything. And this is that contrast thing that I was telling you about. So I see nothing. And then when I come with my ultraviolet light, and look, I'm shining the ultraviolet light at the ceiling. You could see it's purple, but you can't see it, right? You can't see this illuminator in the air. When I shine it on my molecules, all of a sudden I get orange, yellow, green, and lighter green. So thanks, you could turn the light on. So this is how the fluorescence is working. In our bacteria cells, we can fluorescently label things, and we can get really high contrast. Fluorescence is actually all over nature, though, and so I do want to show you a few more examples. Sorry to keep making the lights go on and off. And so fluorescence is really all over nature, and so here's one thing I could tell you about, which is fluorescent tattoos. And again, my kids are in the front row. There are no tattoos. And if we could turn the lights down for a sec. This is some tattoo ink on this black piece. And is it possible to turn the spotlight off? Not so much? Oh, stand over here. Perfect. Okay, so here's some tattoo ink. And here's a UV flashlight. This is very violet light that we can't really see very well. And if I shine it on here, I see that these tattoos are lighting up fluorescently. And so again, these are molecules that I'm giving energy from my flashlight, and that energy is exciting electrons, and when those electrons relax, I'm seeing emission at a different color. So I'm not seeing the purple emission anymore, now I'm seeing the color of these dyes. These things are actually all over nature, and so I want to show you one last demo, and that's these rocks. And actually, in nature, there's lots of fluorescent minerals. And so here's an example that I found online. And these are a bunch of different rocks, naturally occurring. These aren't artificial dyes or anything. And all of these rocks, when they're lit up with UV light or with purple light, they emit fluorescence because of minerals and impurities and holes in the crystal structure. So let me show you some really great examples. And here's the first one. And can we turn that spotlight off for a minute? Thank you. So here's one rock that has some red and green impurities. They're fluorescent impurities. And what I think is amazing here is that this rock, I looked at this in the demo lab a few weeks ago, and I said, well, obviously there's some lichen growing on the surface. This is all mineral. This is not moss growing on the surface. This isn't lichen. So there's minerals inside this rock that light up. And you can see that the illuminator that I'm using here, you can barely see any color. I'm definitely not shining a red spotlight on the rock. Rather, I'm exciting these molecules and letting them emit. And I don't see anything but the fluorescence. So let me show you one other rock, well, two other rocks actually. Actually, I can leave that on. So here's another rock that if I hold up, well, if I turn off the light and hold it to the camera, you could see nothing. That's a good thing. So we see nothing, and this is my background, right? There's nothing in my background. When I turn on my illumination, I see that it glows bright red, and you can see this purple crack at the center. Oops, this purple line at the center. Oh, that's fun. Right here. And so what you see there is a purple line that shows a different type of mineral. And so what I'm doing here is actually two-color fluorescence imaging. I'm seeing two different things. Now, I told you that I'm using a high-energy light, a UV light to illuminate it, and I'm seeing visible light come out. And what's interesting is we can even be kind of wavelength specific. So here's my last rock. And what you see here is the tag. What you see here is basically nothing. And when I turn on the UV light, whoa, right? It's lighting up all green and red. So again, there's two different kinds of minerals in this rock that have really vivid fluorescence. Let me just show you the real red stuff. Have really vivid fluorescence that we could see in the microscope. So thank you, we could put on the lights. So this is to tell you that fluorescence is ubiquitous in nature, and fluorescence is a really exciting way to achieve different colors in our imaging and to see them with no background. Everything that's not fluorescence is just dark. Everything that is fluorescent shines really bright. So this is a picture of my lab over here that was also taken by some great photographers in the College of Literature, Science, and Arts. And what you see here is that we've coupled laser illumination. Actually, you can see it better up at the top right. We've coupled laser illumination, steering around the table, these green and blue beams that you see on the table. We've coupled those on the right side of the image to my microscope on the left side of the image. That's exactly that microscope I showed you that doesn't have leather coating, that's not Hooke's microscope. So in my lab, what we do is we take our bacteria cells and we say, I'd like to see the bacteria cell, and our microscope helps us do that. And we say that we can label individual molecules with fluorescent items, and that also helps us look at the emission. So we have one last problem. So the one last problem is I still haven't told you what we could do about the fact that bacteria cells are just so darn small. So I told you we have a microscope. It gets us down to a micron. I told you that it's hard to see things that are micron. Even though we could see them in the microscope, they just don't have a lot of contrast, so we use fluorescence to address that challenge. But if I want to ask any questions other than, is there a bacteria or is there no bacteria, I'm still kind of out of luck. So if I want to look smaller than this bacteria cell, this is my Vibrio Cholera cell that's about 3 microns in length. If I want to look smaller than this Bacteroides gut cell that's about 2 microns in length, I'm still in trouble. So let me tell you one last technical innovation that really lets our lab look at the nanometer scale inside a cell. And that's thinking about how we can beat this diffraction limit. So remember this figure, I've showed it to you before. What I told you is that even if we have one tiny, tiny molecule, what we're going to see in the camera is that spot that's about half a micron in size. Half a micron, remember, is kind of like the diameter of the bacteria cell. It's pretty big in my books. So the question is, how can we circumvent the diffraction limit? And this is an area I got into. So I was working on optical connects for computers as a PhD student at Caltech. And then I had this opportunity to go to Stanford as a postdoc, as a research scholar, and work in the lab of W.E. Murner, who was thinking about what to do about this problem. And actually, it was very exciting because a few years after I came here, he and some colleagues were awarded the Nobel Prize for this technique that I was fortunate enough to learn and to work on in his lab. And the main idea of this Nobel Prize, this is the Nobel Prize in Chemistry from 2 years ago, is super resolved fluorescence microscopy. And so when you hear the term super resolved, you have to remember what I said about resolved. I said that the resolution was that about half a micron limit that the wavelength of light is imparting. And so the question is, how can we do better, right? Super is better. And the trick that we play, I like to think about this analogy. This is Mont-Saint-Grégoire, which is near where I'm from in Montreal, Canada. And Mont-Saint-Grégoire is a mountain that's kilometers in length, or now that I'm teaching in the US, I say it's miles in length. And Mont-Saint-Grégoire is nice and big, but everyone in this room knows a bit about the shape of a mountain, right? It goes up, and then it goes down. So this is not the best mountain in the world. This is not the biggest mountain, but it is an isolated mountain. It's actually a really interesting geological formation. It's an unerupted volcano. So it actually pushed up the earth. So it's not isolated. So rather than look at the Rockies where you see many, many, many mountains, here we have one isolated mountain. And I'm pretty sure everyone in this room, and you all can try to point, get our exercise for the day, can tell me where the peak is of this mountain. So why don't you all go ahead and point at the peak? Okay, so everyone in the room told me it was somewhere around that middle, somewhere around the peak. And actually, you'd all be a little bit subtly different because this is not a great mountain, right? This is a pretty subtle mountain. So everyone in the room would tell me some place on that mountain with a little bit of what I call an error bar or a measurement uncertainty. But all those measurements would be somewhere around the middle of the mountain. I'm pretty sure that none of you, unless you missed the assignment, told me that the peak was down here in the bottom left corner of the screen, right? We all knew the peak is right up there. And so what I'm doing here is telling you that the peak of the mountain without knowing anything about image processing, we can find the peak of the mountain with meter scale accuracy, even though the mountain is kilometers in size. So if we look at a single molecule, at one dye molecule, we could scale down that trick. So remember, we're coming down this ruler again, and we're coming down to the scale of nanometers, but we do the same thing. So here's the image on the microscope that I showed you already that one of my students took. He put a dye molecule, isolated, alone, on a microscope cover slip and looked at it in the microscope. And here's what he saw. He saw the image of that dye molecule. And so what you see is it's a punctate spot, and it's about half a micron in size. That's that diffraction limit of light. And we do something which is called fancying up the data and then processing it that looks like this. We take this image, and if we turn it on its side, what we see is that it's not just a circle, it's actually a mountain. It's brighter at the center of the spot, and it's dimmer at the left of the spot. So the true data, this is the image I see on my camera, but every pixel, every white square has an intensity, and the true data looks like this mountain in the center on the right. So we have this mountain, and we could do a series of types of algorithms in the lab, and we could find that peak position with really high precision, the same way that you all found the peak of Mont-Saint-Grégoire. So now what we know is that this diffraction-limited molecule actually is at the position of that red dot. We're finding the peak position. So this technique blows me away. This was why I got into this field. I actually read this paper. This is a bacteria cell that they were studying in the Merner lab before I got there. It's called Caulobacter crescentus. And what you're looking at, again, is one single dye molecule inside that cell. It's a structural protein. It's telling us why Caulobacter crescentus has a nice shape. And what you can see is that molecule is a punctate spot, about half a micron, and it looks like a mountain if we process the data. And so we can pinpoint the position of the molecule inside the cell on the nanometer scale. So now, rather than just saying, where is the bacteria cell, we can say, where is that molecule in the cell? And the final exciting development is that because this is all done on the benchtop in my lab, we can actually look at it over time. And so here's that molecule doing a zigzag. If you watch it go left, right, or so from left to right and back, zigzagging across the cell, and I've actually sped this movie up 100-fold, so this is very subtle motion, nanometers per second, and this is saying how do proteins move and work inside the cell. So in my lab, we think a lot about single molecule microscopy, and we do much of what I just told you. We take that molecule, we find its center position, we watch it over time, and we can ask questions like how fast is it moving? Is that motion directional? What part of the cell does it like to move on? Does the motion change as a function of time or position? We can also say if I have many, many molecules, it's hard to see them, but if I have a green molecule and a red molecule, how far apart are they? How does cooperativity work inside a cell? And finally, there's some techniques that I won't have time to tell you about that really go to this super resolution moniker, and that's the idea of making your molecules blink, and it's sort of beyond the scope of what I'll have time to get into now. And in this way, we could find structures with this single molecule localization approach. So in my lab, like I said, we think a lot about single molecule tracking or single particle tracking and what information it could give. So let me take the last 10 minutes to tell you about some of the questions that we've been asking with these methods. And I started off this talk telling you that we have two principal questions. One is, how are toxins expressed by Vibrio cholera? Vibrio cholera, remember, is the bacteria that causes the cholera disease. And we want to know, how are the toxins expressed, with the obvious eventual goal of saying if we understand how the toxins are expressed and regulated, we can actually think about therapeutics to fight them. The other question that we've been asking is, how do the bacteria in your gut digest fibers and all those good things that we're supposed to be eating? So let's start by thinking about Vibrio cholera. This was one of the first projects I started when I got to the University of Michigan. You know, I had been working on model systems, but I was so excited about the big med school across the street and the opportunity to work with more applied biologists who are tapped into these health and disease questions. And what we did was we started looking at Vibrio cholera, and this is a Vibrio cholera cell. Remember that length scale, right? The length is about 3 microns. The width is about 1 micron. So we know a lot about the shape, but that's about it. And the question that perplexed me, that was put to me by my collaborator, Victorita, is how do the proteins inside this cell work together to make the toxin that makes people sick in cholera epidemics? And so what we're doing here is zooming in a totally cartoon way, because all we could do is cartoons, right? Let's go back to my problem and my driving force. And if you zoom into this bacteria cell wall, there's two membranes here, and what you see is that there's these two proteins, TCPP and TOXR, that are on the membrane of the protein. So you can imagine, of the cell. And so you can imagine these are proteins that are expressed in the cell, that can move all around the cell, and that are confined to the perimeter of the cell. And what we went ahead and did was we took the TCPP and we added a fluorescent tag. So now I can look at my cell and it looks totally dark, and I could shine a purple laser on it, and I could turn on this red molecule and watch it move inside the cell. And the really interesting question for me, beyond just specific to Vibrio cholera, is the fact that these proteins have a specific function in the cell, and the function in the cell is to interact with the DNA nucleoid, all the DNA that's inside the cell, and they interact with the DNA to tell it to express the toxin. So the key regulatory position in this cell, in this process, is going to be these proteins on the membrane interacting with the DNA inside the cell. So we set out to look at it, and just to summarize a bit what we did, my student Beth Haas was working on this project, and what she was able to do was track molecules. And so each of these little blue and yellow squigglies here is the motion of one TCPP protein inside the cell. And so what she could do is track that molecule. She could confirm that indeed that molecule does stay on the perimeter of the cell even as it interacts with the DNA inside the cell. And then we started to probe this interaction between TOXR and TCPP by making knockouts. So basically, the types of approaches we think about in my lab are, how does TCPP move? It's supposed to bind to TOXR. If we knock out that putative binding partner, does it move faster or slower? And that tells us something about the interaction. If we knock out that site on the DNA it's supposed to bind to, does it move faster or slower? And overall, what we found was a new mechanistic model for how these proteins interact. And this summarizes it. And again, beyond the scope of what I'll have time to describe today, the main idea is that TOXR and TCPP work together to bind DNA, but they work together in a sort of interesting model where TOXR leads TCPP to its target. So that's one of the stories, and ongoing work in my lab is still thinking about how does now TOXR move, and how does the DNA move, and how are they all finding each other? Because I think these are very interesting questions for cholera, but also questions in general about having big picture paradigms to explain how bacteria cells work. On the other hand, we have a project with our collaborators, Nicole Koropatkin and Eric Martens, and that's asking the question of how the bacteria in our guts digest complex carbohydrates. And so let's go to these cells. The cells that we're thinking about here are Bacteroides theta iota omicron. It's a mouthful. We call them B theta. And the B theta cell, much like Vibrio cholera, has a membrane. And if we expand the membrane, what you see is that there's an outer membrane and an inner membrane. And there's proteins on the outer and inner membrane. So the membrane of the cell maintains the cell shape. It lets the ions balance out. But also there's proteins on the outside of these cells. And the interesting thing is that proteins on the outside of the cell are always used to recognize what's outside, to do import, to regulate function based on those types of recognition events. So these proteins on the surface of the cell, they're called SUS, which is starch utilization system. So they are the proteins that help the bacteria to utilize starch. And what these proteins do is they recognize sugar molecules, different types of starch, carbohydrates, fibers in the cell. They bind them in this cartoon. These little bite-sized pieces are binding sites. They break them down into smaller units. And then SUS-C is this pore that imports the sugar into the cell to feed the cell and ultimately feed us. So things like fiber that we can't ourselves digest, these guys can do it for us. What we're really interested in is Sus-G as a starting point. Sus-G is this red protein in my cartoon. My collaborator, Nicole, is a structural biologist. She actually was able to go to an X-ray facility and solve the structure. This shows you where every protein is in the Sus-G protein. So we know a lot about this protein, and then we can do our tricks. So our main trick is, of course, label it with a fluorescent protein or a fluorescent tag. And now what you're looking at over on the right are two B theta cells, and you see a couple little white dots up at the top and up at the bottom. And those white dots represent single Sus-G molecules on the cell. And now we can ask the same question. So we can watch Sus-G move in the cell. So what you're watching here is a real-time movie that shows Sus-G exploring the surface of the cell. And you all know what it's looking for, right? It's looking for starch. So Sus-G is exploring the surface of the cell. Even if there's no starch around, it's diffusing freely around the surface, looking for starch. And again, we could track its motion with these lines. But the real question is, how does Sus-G interact with sugar? And so what my postdoc, Krishanti Karunatulaka, who was working on this project, did was she went ahead and she said, okay, I have these cells, Sus-G, that enzyme on the surface, it's labeled in red. I'm going to feed it some starch that glows green. So the starch is now labeled in green. And now in the microscope, she can actually watch the red and the green, much like with that rock. We saw red and green fluorescence both. And what she sees is this. Here's a panel at the top right of the same B theta cell. On the left, you see the position of the starch, and we pointed it with a yellow arrow. On the right, that's the red channel. What we're watching is Sus move. And what you're going to see is that some of the Sus lingers at the position of this yellow arrow at the bottom, while other Sus proteins continue to explore the surface. So here's the movie, and what you see again is the Sus moving. Krish has put tails onto these tracks, so what you see is that the white data, that's raw data. That's what we collect in the lab. And then those green and red thin lines that you can see on top of them, that's the trajectory. So now we can ask questions like, how does Sus-G explore the surface of the cell, and how does it respond to the sugar? So overall in my lab, what we've been thinking about is single molecule imaging as a very sensitive and very small scale approach to asking questions like, how do different proteins work together? And we're really fundamentally interested in questions about disease and about health and general guiding principles of how bacteria cells work based on this biophysical approach. So let me finish by acknowledging my group. Myron started by saying that there were a lot of grad students in my lab, and it's true. And you all have probably heard that the job of a University of Michigan professor who runs a lab tends to be less sitting at the microscope, and I don't do a lot of that anymore. These are the guys and girls who do that work. So these are my students. These are the junior lab members. They're also sitting at the front of the room. And so my students, my graduate students, undergrads in my lab, and postdoctoral scholars in my lab have done a lot of the work. And the other thing that I alluded to is that a lot of this work is highly collaborative. We're really lucky to have great collaborators. The Vibrio Cholera work I was telling you about, we do with Victor Rita, who's at MSU. And the B theta work I was telling you about, we do with Nicole Koropatkin and Eric Martens. And there's other bacteria work. I know many of you were here last week when my colleague Lyle Simmons gave a talk. We're also thinking about those DNA repair processes that he mentioned, and he figures up in here too. So I'm really happy to take any questions, and I appreciate you all coming out this morning.